Hello everyone. Today we are going to dive deep into the fascinating topic about the world of immunology. We'll be focusing on the soldiers of our immune system, which is also known as immunoglobulins or antibodies. So here in this section, in the next approximately 20 to 30 minutes, we will break down this complex structure, the complex architecture about the immunoglobulin, the structure of the immunoglobulin. We'll understand how they are classified, what the types of immunoglobulin, and also we'll explore the unique functions which are under these immunoglobulin types. So to understand our immune defense, to understand about anything about immunoglobulins or antibodies, we'll first need to know what is immunoglobulin. So you see, immunoglobulins or antibodies, what are they? They are Y-shaped glycoprotein structures which are generally produced by plasma cell. As from the previous lectures, you guys will be knowing that the helper T cells, what they do, they will activate the B cells or the B lymphocytes that will further activate the plasma cells, and these plasma cells will secrete, or this plasma cell will produce the antibodies. And these antibodies or immunoglobulins, what is their primary function? The primary function is to identify and to neutralize or to kill the foreign invaders or the antigens, the foreign pathogens like the bacteria and the viruses. And apart from this, you also remember the constitution, the constituent of this immunoglobulin. They are approximately you can say 20% of plasma proteins. They are actually present in 20% of approximately 20% of all the plasma proteins in the blood. So now we'll look into the structure of immunoglobulin. So here in the first slide there is a structure. In the second slide I will be showing I will be showing the structure in whole slide, and this one is like the introductory of this structure. So I have written here immunoglobulins. So basically in the structure, let's actually go to this structure perfectly. So let's go to the structure perfectly in the next slide. So look, this is the structure of a typical immunoglobulin. So basically, if you look at the structure, you will see this as a Y-shaped structure. You've seen this structure. This structure is Y-shaped. In the above portion, there is two arms. In the lower, there is a stem. You see that there will be a Y-shaped structure. So this structure is basically comprised of 4 polypeptide chains, and out of these 4 polypeptide chains, two identical heavy chain will be there, and 2 identical light chain will be there. You see the green colors chains, which is the lighter green one are the heavy chains already mentioned in the structure, and the darker green one are the light chain. So why it is named as heavy and the light? Because of its molecular weight. High molecular weight is considered as the heavy chains, and low molecular weights are considered as the light chain. So these chains, heavy and the light chains, all of them they are held together or glued together by disulfide bonds. Disulfide bonds are covalent bonds which will help or which will glue both the heavy and the light chain. There will be also interchain disulfide bonds among the heavy chain itself or the light chain itself. So the structure will be, the chains will be brought together by these disulfide bonds. So now we'll go to the regions of this particular immunoglobulin structure. So the regions here it is actually divided into 2 regions. One region is the variable one, or the variable, you can say variable region or variable domain, and the other one is the constant domain or the constant region. So you see in the variable region, if you see the variable region, it is actually situated at the N terminal portion. It is actually situated at the N-terminal portion. And if you see the constant region, mostly it is situated at the C-terminal portion. And the C-terminal is actually there where the two heavy chains are present. But in case of N-terminal, both the heavy and the light chain will come up. So in case of that variable region, why it is known as variable region? Because there will be variations. And what variations will be there? There will be variations in case of the structure. At the variable region, there will be variations in case of the shape, so that the antigens bind and fit to it, just like a lock and a key. And in case of constant regions are the lower arms, and means the lower one, the stem one. Also, it is also present on the upper one also. So actually, mainly it is present in at the C terminal portion. So it will be relatively same among the all the within a class or all the class. It will be relatively same only. So what it does, it will determine how the antibody will work and how it will interact with the rest of the immune system. So that's about the regions, variable in the this one. You can say the constant region. So there are also fundamental segments. Fundamental segments like the Fab segment, which is also known as the fragment antigen binding. Fragment antigen binding are two arms which are responsible for antigen binding. The Fc fragments, which are also known as fragment crystallizable. Fc is also known as fragment crystallizable. That is the stem of the Y shape. Y shape is immunoglobulin. What it will do? It will handle the biological activity mediation such as binding to complement proteins or the cell receptors. And lastly the region or the segment, if you have seen in here in the structure, in the dog leg structure, in the sharp turn or between the Fab and the Fc region or the fragment crystallizable region, you see a sharp turn is there. In the sharp turn, the amount of disulfide bonds will be more, or the concentration of the disulfide bonds will be more. So why the concentration is more? Because it will provide stability to the structure, and that sharp turn and that portion is known as considered as hinge region. Already mark here that sharp turn is considered as the hinge region. At the hinge region, the concentration of disulfide bonds will be more, and this hinge region will provide flexibility to the flexibility to the Fab arms. Fab arms mean the two upper arms. The upper portion, in the Fab portion, here are the two upper arms are there, right? So it will provide flexibility to that upper arms so that it can move and it can reach the antigens in the variable surfaces of the cell, and it can attach with the antigen or it can reach the antigen. That's it. So this is all about your this one immunoglobulin structure. So now let's come to the different types of immunoglobulin. If we come to the different types of immunoglobulin, we'll find like they are basically classified into 5 types. The first one is the IgG, second one is the IgM, third one is the IgA, and fourth one is Ig, and the fifth one is IgD. So basically, if you see the structure here, if you see the structure of the different types of antibodies or different types of immunoglobulins here, they are basically the IgG, IgE, and IgD. All of them are monomer, means they are having only one Y-shaped structure. But in case of IgM, they are having 5 Y-shaped structures, so they are considered as a pentamer, which are joined by the J chain. They are connected by the J chain or the joining chain. And IgA is a dimer. Actually, IgA also can be monomer, but when it is secreted, it is a dimer. So why dimer? Because they are having 2 Y-shaped chains. And also remember this, like if there is it is a monomer, it will be having two arms, right? It will be having two arms, so it will be having two antigen binding sites. If it's a pentamer, it will be having 5 arms, means you can say 10 arms. For one Y shaped it will be having 2 arms, so for 5 Y shaped it will be having 10 arms. So 10 arms will be having how many, you can say how many binding sites? It will be having 10 binding sites, and IgA will be having 4 binding sites. So you have to remember these things. So you see the IgG, it is also known as the abundant protector. If you see at the prevalence scenario, it is present at it is approximately 80% of the total serum immunoglobulins. IgG is approximately 80% of total serum immunoglobulins. If you see the lifespan here, it is the longest of all the classes. It is having 23-day half-life. Okay, a molecular weight is also there. It is around 1,50,000 Dalton. And there are also subclasses. We will not go to the subclasses here. So the main function, the primary function of IgG is it crosses the placental barrier. This is the only immunoglobulin structure, this is the only antibody structure which can cross the placental barrier inside the mother's womb during pregnancy. So if you consider just like a mother that can pass her own immunity, directly to her unborn child at the womb itself to protect them from various, you can say infections and other things, and also to give them and to protect them in the first few months of life. So this is about IgG. So next we will be considering IgA. IgA, it is also said here as the mucosal sentinel. Why it is considered as the mucosal sentinel? So because it is the master, or it is considered as the master of external defense. So if you see at the prevalence here, it is around 10 to 13% of serum immunoglobulins. It can actually exist as monomer, but when it is secreted, already I have told, it forms a dimer linked by a JSN polypeptide. In the structure also it's been shown here. So its half-life is around 6 to 8 days, and its location is, it is highly present, its concentration is highly present in the mucous membranes, in the saliva, in the tears, and in the first mother's milk, which is also known as colostrum. And its main function, as I have already told, it's a master of external defense. It's the first line of external defense. It will trap pathogens in the gut or the respiratory tract before they can enter the bloodstream. So this is all about IgA, and apart from that, you have to remember that IgA is a dimer which will be having 4 antigen binding sites. So the next one is IgM, the early responder, the first antibody which is produced in a primary immune response. If you see at the structure, it's a pentamer, 4 Y-shaped structure will be there, means 10 arms will be having 10 antigen binding sites. Prevalence, if you see here, it comprises of 5 to 8% of serum immunoglobulins. Half-life is less, 5 days, and you know, since it's a pentamer, it will be having a maximum amount of this one, the molecular weight will be really high. It will be a heavy molecule. So it's a first antibody that will be produced in a primary immune response, and its main function, since it's a pentamer and it will be having 10 binding sites, so it will be absolutely most efficient in case of agglutination, which is clamping of the germs or clamping of the antigens together. and it is a first antibody that will be produced in a new infection. So this is about IgM, if you say, and it's considered the early responder. So next one is the IgE, the anaphylactic trigger. So if you go to the prevalence here, it is found in trace amounts, less than, I can say, 1%, around like only 0.002% of normal serum. Half-life is also really less, 2 to 3 days, and molecular weight is around 190,000 dalton. It is a heat labile, you can say, antibody. It will get inactivated at 56 degrees Celsius in 1 hour. If it gets more heat, it will not be, it will get inactivated. And mainly its function is a primary mediator of this allergic reactions, asthma. It will bind to this mast cell and the basophils. When it will sense an allergen in your body, it will directly trigger the release of chemicals that will cause sneezing or inflammation. So it is also considered as the primary defense against any sort of parasitic infections. So this is about Ig. It will be a monomer. Mainly it is, again, it is for, its main function is for allergy and allergic reactions. So this is the last one. This is the fifth one. You can say the immunoglobulin, that is IgD, also known as the cellular receptor. If you see at the prevalence, it's really less. It's less than 1%, 0.2% of serum. Half-life is also really less, 2 to 3 days. If you see, it's a monomer. Actually, it is also a monomer. So its location, if you say where it will be present in the body, it will be present exclusively, most of the time, almost exclusively, it will be present at the surface of B cells. It will be anchored. It will be anchored at the surface, anchored to the surface of the B cells or B lymphocytes. And its main function is it acts like a recognition receptor. Means you just think of like it as a sensor that actually that will tell about the B cells when it has finally found the like you can say the specific germ or specific antigen. What it will do? It will signal the B cell that I have found this particular antigen which I'm supposed to fight against, and I will initiate the entire immune cascade. So IgD is all about that. That's why it is known as the recognition receptor. So this is all about IgD. I hope that you guys have understood the types of these immunoglobulins comprising of IgG, which is you can say the it can cross the placental barrier. It is the primary response for the newborns, or you can say the unborn child. If you consider the IgA is the first line of external mucosal defense or external defense. IgM, it is the first antibody which is produced during new infection. It has roles in agglutination, clamping of the antigens. IgA, it has role in case of allergic reactions. And IgD, which has role in case of, you can say, it has role as a recognition receptor, which can recognize a particular antigen and it will signal the B cell and it will initiate the entire immune cascade. So our body, it doesn't only use one type of antibody or immunoglobulin to fight the infection, but it will consider, or it will use for the following 5. The shown that I have shown you, the 5 types of the antibodies or the immunoglobulins. It will use all 5 of them. It will use the longevity of the IgG, the breadth, since breadth of IgM, since IgM is a pentamer, the barrier defense of IgA, first line of external defense, the sensitivity of IgE, it has roles against allergic reactions, and the detection power or the recognition power of IgD. And all together, what they will do? They will make sure that no matter how an invader enters or how an pathogen enters our body, our immune system is ready to respond and act against it. So I hope you guys have understood this immunoglobulin topic. I've covered the basic of immunoglobulin, the structure of immunoglobulin, and also the types of immunoglobulin. Thank you so much, and sorry for being late. I was actually busy on my personal stuff also, and in case of my like study stuff also, since my finals are approaching, so I'm not being regular enough. So sorry for that, and I hope that you guys have enjoyed this lecture. Thank you so much.